Okay. I was thinking about, what Paige had suggested for today, and that is to go over the muscles, which we could still certainly do. But I got thinking more about that, and I guess I have a couple thoughts. One is, because this is designed or designated lecture time, we should certainly first talk about questions related to lecture. And then if we've exhausted that aspect, we can talk about lab. Also understanding that we do have a designated review time scheduled, which I don't know if all of you are signed up for that, but we can use that as well as time to review lecture or lab stuff. So I'm a little hesitant to say, let's take lecture time and do lab, especially if there's questions related to lecture. The other thing that I was thinking about was the fact that last semester, or last spring actually even, I posted some videos that went over the torso, leg, and arm models. So I wanted to go back and review that and remind you that those existed. And I'm trying to remember if I posted these on the current semester Blackboard course shell. So I'm looking as we speak to see if those videos are there, because that might be something that you should be aware of too. Oh yeah, okay, so let me share my screen. So do you see the Blackboard stuff here? Okay, so in the folder entitled Stuff to Copy and Bring to Lab Each Week, if you go down to muscle identification videos, corresponding to exercises 20 through 23, which is what we just finished up today. And you click on that. There are several videos here, one on the face and head muscles, one on the abdominal and chest muscles, one on the, actually two on chest and the abdominal muscles. And then we've got an arm, and we've got actually two arms, and we've got a couple legs. So I wanted just to let you know that these are here. And when I put these together last spring, when we had to go totally remote, I chose those videos that had the same exact models that we have in lab. So, you can take a look. These look familiar to you, right?
That's really helpful. Thank you.
Yeah. Now this one, as I look at this looks like a little different coloration of the muscles. So this could be from a different manufacturer, but this arm is the same. This may or may not be the same. I just don't recall. This is the same. So I just remembered that I had these. And so, this could very well substitute for what we would have done. And we can still do. I'm not saying I won't do it, but if they're here, you can look at those too. I bet you didn't know they existed in the course shell, did you? Yeah, because I didn't stress it. You should definitely take some time and look through Blackboard course shell, because this is stuff that I may forget to say to you because we're meeting face to face. Why would you want to look at a video? Well, certainly you might want to look at a video to review for a quiz or a practical. So yeah, I get that. The same applies. Trying to think now, respiratory organs. Oh yeah, do you see the Blackboard thing on your screen? So here's a model of the larynx. Here's a model showing upper and lower respiratory tract structures in a torso. This is not the same torso that you have, but it's somewhat similar. So go in and take a look what I've got here. Might be other important stuff here that you might find helpful. Cardiac cycle, when we get into that, when we get into the heart. Yeah, so there's some nice videos in here, and they're all available for you to look at. Okay, so I just wanted to mention that. So as I said earlier, we should be starting the respiratory chapter this week if we attempt to adhere to the lecture schedule. We've got this whole week devoted to respiratory system. We have the first half of next week. Again, check the syllabus. We start blood next week and carry on from there. So all of the PowerPoints, all of the video lectures, I think I've freed up for the rest of the course in Blackboard. Has anybody started looking at respiratory system at all yet? Freya says she has. Okay. But a lot of you guys probably are.
I really kind of gave like a reading review. I didn't study as thoroughly as I would have.
Sure.
I went over it.
Okay, that's great.
Like it's easier then when you talk about it to like connect things if I go over it first,
Oh yeah, absolutely. No, that's good. So today's stuff that we did, you were not totally unaware of some of those structures probably.
No. Yeah.
Yeah, very good. I assume the rest of you guys are in the muscular system chapter then, number 9, probably. Yeah. So let me just open up the floor. Are there any topics, in the muscular system chapter you'd like to go over or review?
So I guess this is sort of a similar question I had when we were talking about aerobic and anaerobic.
Uh-huh.
As far as the muscular goes, I mean, obviously that's connected. How long? I guess I was just curious, like how long does that like the lactic acid, like the oxygen, does that have to do with like how if you're in better shape, does that oxygen deficit, is that less time?
Yes. So let me try to determine what you're seeing here. So if we go to the PowerPoint, which you should see in front of you.
Okay, yep.
And if we scroll down to that slide that talks a little bit about oxygen debt. Okay. And that's this. And I don't know where you all are in chapter 9, but this is about, I don't know, more than halfway through the chapter. The idea here is if you overexert yourself and you deplete your initial ATP stores and you deplete the creatine phosphate stores and you tap into aerobic respiration and you find you're exercising quicker than the mitochondria can keep up in terms of providing the muscle with ATP, and you get to a point where your cells have to go anaerobic because you can't get adequate oxygen via the bloodstream to the muscle. They go from the aerobic state to the anaerobic state. You might remember aerobically cells can generate somewhere in the neighborhood of, let's say, 32, 34 ATP per glucose.
Right. Okay.
Anaerobically, how many ATPs can a cell generate per glucose?
What was it? It was like 56 or something, or I don't know. No, that was being smart.
Anaerobic.
Oh,
Right. Isn't it like 4, but then you use 2, so it ends up just being 2?
Yeah, you come out ahead
okay.
two. So
That's
it's like
quick.
it's like 1/17, 1/16 as efficient. Okay.
Okay.
And at that point in time, when your cells go anaerobic, by your cells, I'm talking primarily muscle here, then the body begins to build up what's called an oxygen debt. And that's what Freya was kind of getting at when she asked that question. And so here we see a scenario whereby 3 athletes have reached the wall. They've reached the wall and they've hit the wall. You know what I'm saying? They mentally may want to finish the race, but the body says, I don't care what you want to do, you're not going to finish the race.
Well, it just does it shut down? Is that when you kind of almost like?
Yeah. Well, these guys are, these first 2 guys are hit the wall. I mean, they have hit the wall. They aren't going anywhere.
Right.
Right. And this guy looks like, well, who knows? Hard to tell. But these guys, he's on the ground. He's ready to fall down. You know.
Right.
This
Well,
guy
so-
should not be running the race. He needs to be sitting home having a beer on the
If
floor.
you're in better shape, though, will it take less time for that oxygen deficit to be?
Yeah. And why is that? Why is that? Because when you are training to win an Olympic gold medal in speed skating, or
Right.
if you are a thoroughbred racehorse, you're genetically predisposed to perhaps winning than if you were a Clydesdale. So I got to think about the genetic end of this too.
Right.
But let's just look at the overall idea of athletic training. Why do athletes train? Well, we know they train because by training, they increase their odds of being successful, right? But
Right.
what really happens physiologically when you train? And Sadie, I know you work out. You told us that. You're out there working out every day, right? Or every couple of days. What that's providing for, of course, is a lot of benefits. I don't know if you're an athlete or you like to play soccer or whatever, but if you were training to, again, perform at some high level of competition, then what we find is that the more you train, the more glycolytic enzymes your muscle cells make. Now, what do I mean by glycolytic enzymes? Well, what's glycolysis? It's that process that we said occurs in the cytoplasm of the cell when glucose is degraded into 2 pyruvic acid molecules.
Right. Okay.
There's 10 different reactions, 9 or 10 in that series of glycolytic pathways. So you make more of these enzymes if you train. Why is that good? Well, that means you can undergo glycolysis much better than if you didn't have as many enzymes.
Right.
I mean, we all have them, but you have more if you train.
Okay.
Also, you increase the vascularity of muscle tissue. Angiogenesis, we call it. The synthesis of new blood vessels, i.e. capillaries. Why is that beneficial?
Surface area. I'm just kidding. I threw that out there.
Well, it is increased surface area. You're absolutely right. But give me some specifics. How is this helping you as an athlete?
Well, just I guess the more surface area there is to absorb it, the more
Absorb what?
will absorb the ATP.
No.
No. I'm
no.
sorry, I'm confused.
That's being made in the cells.
Right.
It
Okay.
allows more
What's
oxygen.
blood do? Go ahead, Paige.
It allows more oxygen to get to the muscles.
Exactly. Your bloodstream is the highway interstate system that's delivering the oxygen to the tissues. Why is getting more oxygen to the tissues beneficial?
Keeps them alive, right?
Well, yeah.
I mean, it keeps them going, functioning, living. Well, and the oxygen.
Yeah. What's the oxygen provide? Well, it provides the cell, if it can stay more aerobic, it can crank out more energy. Right? Just think about it. If you have, what's a good metaphor? It's as simple as if you have more blood traveling to the muscle, you have more oxygen being delivered to the muscle, and the muscle can maintain its aerobic status longer and reach oxygen debt later. So it gets to the heart of your question. And we find that the number of mitochondria increase in muscle fibers when you train.
Oh,
I hope you see the obviousness of this, if that's a word.
yeah. Well, so okay, so everything's more efficient, really.
Yeah, sure. Right?
Yeah.
But there is some limit to this. And that gets at the last bullet here. And it's sort of hard to explain in terms of humans, but there is this concept that you can train as hard as you want or as long as you want or with the best positive mindset that you can have, but you still may not perform as efficiently as someone who is genetically better predisposed to run a race, play basketball, speed skate, whatever it is, whatever the sport is. Some people have the genetic predisposal that puts them at a benefit over others. It's a genetic thing.
At a younger age, would that help with that?
Well, sure. You know, you don't see a whole lot of 78-year-old linebackers.
Right.
Right.
Right. I mean, I read once that by the time we're like 4 or 6, that we lose like 60% of our flexibility, and if we could maintain that, it would like increase our athletic ability incredible, you know, like so much. So I just wondered if there was any other time where that was like sort of open, but I guess it's more has to do with when you're young.
Well, each chapter, at the end of each chapter, there's a section called, what is it?
I saw that. Okay.
Lifespan changes. I think it's what it's called.
Yep.
Right? Lifespan changes. And that's getting at what happens to each organ system as you get older. So, you know, that's just an age-related thing. But what I'm trying to get you to think about in this fourth bullet is that some people just have innate talent because of the genes they inherited from their mom and dad that might make them a better basketball player than somebody who really wants to be really, really good and be as good as this fella, but just doesn't have the genes. You know?
How much do you think like a combination between like good genes and like a good lifestyle would benefit?
Oh yeah. Oh sure, absolutely. Yeah, sure.
So like with the athletic training, because we were talking about like with the slow twitch and like the fast twitch, like red fibers,
Yeah.
and then like
Yeah.
white fibers. So, like, how does that play into this? Because like you said, like marathon runners generally have more red fibers, and then like sprinters generally have like white fibers, which is like more fatigue.
Yeah. This gets exactly at what I was just talking about genetically, because the percent of slow to fast twitch fibers, from my understanding, is genetically determined. Now, I don't know what the ratio or difference is, but it doesn't probably take a whole lot of difference that might make you better predisposed to being a sprinter. Because as you say, you have a higher percentage of fast twitch fibers versus a marathon runner, as you said, that has a higher percentage of slow twitch versus fast. We have both, but it boils down to, I don't know if percentage is the right word, but the relative amount of these fibers in one's
Based on
body.
where they are, you mean percentage wise?
Yeah, I'm talking about in your leg muscles. Let's say if you're talking running.
Well, so nervous of being I don't want to offend anybody, but don't just because of evolutionarily, like African American or whatever the heck people want to call them, don't they tend to have more fast twitch muscle than a Caucasian?
I don't know I'd have to do research. I don't have any sense of that.
I did
I
a
don't know.
paper a long time ago about it, and we just talked about how like the evolutionary of like slaves and like the way they worked and stuff comparatively,
Well,
which kind of made sense to me.
yeah, yeah. Well, no, there's true. The evolutionary process is at play, and it affects us as well as it does a tree or an insect or whatever. You know, we have to understand the basic
Level.
evolutionary. Yeah, and so somebody who lives in the high Arctic versus somebody who lives near the equator are
Right.
going to have different physical attributes because of where they live and the conditions that they're living within.
Right.
That's just the reality. That's biology. And that makes that individual more likely to survive in those particular conditions.
It's awesome, isn't it? I mean, I actually had a class that was about or I taught a class about just trying to like objectively look at the differences of the physical parts of race and why they are that way. And you wouldn't believe how many people today, like, it blew their mind away. Like, just the melanin, I mean, just things like that. They didn't even understand it.
Well, exactly. It's a function of a little bit of knowledge, biology, little A and P, and also being cognizant of the fact that you got to be kind of careful how you discuss some of these things without coming across as being, I won't say racist, but people have all sorts of interesting attitudes about all sorts of things. But we can have a discussion, a conversation about this. Yeah.
There's that cancel culture now. You can't say anything.
Yeah. So anyway,
I have a quick question on that real quick. Can we kind of influence which type of fibers we have more of by the type of training that we do? So like, say I was a sprinter in high school for track, and like if I decided as an adult to start running marathons, like would my body develop more red fibers, or am I just kind of genetically predisposed one way or the other?
I don't know the answer for sure, but my guess is that you are not able to convert slow into fast and fast into slow. But I have not done the research. I would think it's more genetically determined.
Okay.
That's just my gut level reaction. I'm not basing
So
that
like
on any facts.
distance runners then, because so like with track we like weaned out like the sprinters, mid distance and distance. So like would like mid distance runners have like a good mixture of both then? So like they can do both?
It's possible. Exactly right. Yeah.
Because like I did some sprinting things, and then I was also mid distance, but then I also did distance. So like I did like a little bit of all of them.
But did you excel in one versus the other?
Yes, I was better at like mid distance than sprinting, but like I can still run like the 5 miles for like distance wise though.
That doesn't mean, I'm not saying that a sprinter can't run a marathon and a marathon can't be a sprinter, right? We can all do those things. But if you look at the data over the long term, there are some people, as you say, Sadie, that are better mid-distance runners and others that are better sprinters. I mean, it just goes to reason. If you think about the Olympics, people train, but are also better able to perform at that particular 100-meter dash or whatever they call it. I don't even know what it's called anymore.
Yeah.
Versus the 5K or the what's the I love that skiing shooting sport. What's that called?
Ski?
No, on the skis and they're shooting targets. Remember? Olympic winter sport, biathlon. Anyway.
You have no idea how much relief this conversation has given me. Now I can say I have a reason why I can't run marathons.
There you go. Okay, so how about other questions on muscular system?
The motor units. So like I just don't know if I'm like grasping it correctly.
Yeah.
Because like there was no audio on this video. So is it basic? Like the audio.
There's no audio.
Yeah. Like
On-
it like came in like spurts, but then like the whole like last 10 minutes of the video, there was no audio.
Really?
Yeah, and it like would just like cut out. So is a motor unit basically like a bundle of like skeletal muscles that are like controlled by like a motor neuron?
That's exactly what it is. So you
Okay.
see on your screen there the definition. Here's the sketch. So we have two motor neurons shown here in yellow. This first motor neuron is synapsing with two muscle fibers, right? Actually, no, three. We got 1, 2, and then here's a branch off to the third. While the second motor neuron is synapsing with just 2 muscle fibers. So this brings up this concept that you can have motor units that all consist of a single motor neuron, but can synapse with a number of different muscle fibers. Some motor units might only synapse with, say, 10 or 11, 12 fibers, while other motor units may consist of a motor neuron that synapses with several hundred muscle fibers. And the difference is that if you synapse with fewer muscle fibers, you have more precise control over the action of those muscles. For example, if you look at the muscles that result in you moving your eyes up and down and over and all that stuff, right, as you're using right now to look at the screen or taking notes, those muscle fibers that you're contracting to move your eyes, each of those motor units consists of a relatively small number of muscle fibers that they innervate. Compare that to big, broad postural muscles of your back, the latissimus dorsi, for example. There you've got motor units of a single motor neuron synapsing with 200 or 300 muscle fibers, giving you more coarse action, more coarse movement than you do, say, with fingertips or eye movement. And I think I say that, or at least I thought I audioed that in the lecture. It's described somewhere in the book too. I'm kind of concerned that you're not getting all the audio. I'll have to go back and check out that Zoom lecture because I didn't realize there was audio gaps. So this has implications. So do you want to talk about the physiology stuff here? Okay.
Yeah, because this was all the stuff that was muted.
Really?
So I just like read more so like in the book about this part and like just tried to like grasp it from the book more.
Okay, well, I'm sorry because I was unaware that there was an audio. So let's talk about this. So again, important to define a few things. We already defined motor unit a few moments ago, which was again a motor neuron and the skeletal muscle fibers it controls. That's a motor unit. Here's another concept that's really important, referred to as threshold stimulus. Now, in the olden days when I would teach a lab on this, we used to take frogs and we would use the gastrocnemius muscle of the frog. We would double pith the frog, and I won't go into the gory details of how that happens, but you basically scramble the brain and spinal cord of the frog, kill the frog, and then you dissect out the gastrocnemius muscle. And you know where that is? From this morning. Remember gastrocnemius, calf muscle? And you hook that up to a machine and you stimulate it and you measure the amount of contraction that the muscle undergoes upon stimulation. And so if you had an isolated muscle fiber, okay, not an entire big muscle like the gastrocnemius, but let's envision a single muscle fiber, but the same actually applies to the entire muscle, and you delivered a stimulus, an artificial stimulus to that muscle with a stimulator machine, you'd find that there's a certain stimulus that you'd have to achieve, certain voltage you'd have to achieve before the muscle fiber would react, i.e. contract. And if you stimulated at a voltage below the threshold, the muscle fiber would not contract. It's only if you reached it or exceeded the threshold that the muscle fiber would contract. So now let's expand our thinking a little bit, remembering that a motor unit is a single motor neuron and the muscle fibers it controls. So each motor neuron also has its own threshold. If you deliver a stimulus to that motor neuron, which is connected to the muscle fibers, at a sub-threshold voltage, the muscle fibers it connects to or innervates will not contract. You've got to reach the threshold or exceed it before you will induce contraction.
So with like this, with like voltage and stuff, like basically like when your heart stops working, those like things that they do and they like do to your chest, is that like the voltage? Because like isn't like your heart like cardiac muscle? So if it stops working, is this like the stimulus they're trying to get to make your heart like start going again?
Yeah. When you take somebody and you get the paddles and you say clear and you go kaboom and the guy jumps out of the bed right off the gurney, that's what you're thinking about, right? Starting the heart up again.
Yeah.
Yeah. So somebody who's experiencing that, we would say that his or her heart is fibrillating. That's the term they use, fibrillate, meaning it's quivering. It's not pumping and contracting and relaxing like it should. If you looked at somebody's heart on the operating room table and the heart goes into fibrillation, it would look like it's quivering in the body. That's not good. So at that point, you would get the paddles and you would put them on either side of the heart and you would deliver a stimulus to try to defibrillate the heart. So those shocking, what do they call them? The suitcases that are around the campus, the defibrillators.
The AEDs.
Yeah. So you're delivering a stimulus to the heart to defibrillate the heart so that it goes from the quivering action to the more normal pumping process.
Okay.
This is a little bit different. It's still measuring electrical activity. Like when you have your EKG done, you're measuring small little voltages that are being picked up by sensors on your skin, whether it's the 12-lead chest monitors or the leads, or you can kind of put them on your wrists and arms and legs too. You can get an EKG that way. So it's kind of related, but it's a little bit different than this. We'll get more into the heart stuff coming up later in the semester.
Would this be kind of like when they go to test your reflexes at your annual physical, like if they don't hit your knee hard enough, it's not going to stimulate that response, like that kind of thing?
Well, when they do reflexes with the reflex hammer and look for the leg to extend out, the old classic,
Mm-hmm.
What you're doing there is you're mechanically hitting a certain nerve plexus that induces an impulse that causes the muscle to contract and the leg to extend.
Mm-hmm.
So it's kind of similar, but the actual mechanism of inducing a reflex at the doctor's office, he or she is usually mechanically hitting a certain area where he or she knows there's a cluster of nerve ganglia or complexes. So it's head related, but that would not happen unless he or she struck the right spot.
Mm-hmm.
But you're right, that would trigger an impulse and would trigger a contraction.
Mm-hmm.
Yeah. This is a little bit more theoretical in that we're stimulating this muscle artificially. Let me see if I can just show you a picture, if I can find it real quick, of the setup, so you have some idea what I'm talking about. I'm not sure if I'm going to be able to find something real quick here, but I'll try. Okay, let me share this screen with you. All right. I'm looking for something similar to what we did. When I was in grad school, I did a ton of these experiments. Let's see, that's not a good picture. So basically it looks like this. I don't know, can you see this? So here's the frog. Again, you have to, how shall I put this? You have to immobilize the frog. It's not feeling any pain, but its muscle tissue is still viable. And you put various electrodes here into the gastrocnemius muscle of the frog, and then you stimulate it, and you can measure the response. Okay, that's in essence what we're talking about.
So is that like electroshock, like therapy? Like, you know how some athletes do that?
Yeah, it's sort of like that,
Okay.
right? Yeah. I just don't see a good setup here that I can show you. I'm surprised there isn't one better shown here, but there isn't. Okay, so back to the diagram. Okay, do you see the PowerPoint slide again now? Okay. So now we have to think about a muscle of our body. Let's pretend gastrocnemius muscle. Okay. But this applies to any major muscle group, any major muscle of your body, any of those that we've talked about this week or last week. Biceps femoris, triceps brachii, latissimus dorsi, you name it, this applies. Skeletal muscle. In any of those muscles, you have many motor units. Now remember, what's a motor unit? A motor unit is a single motor neuron and the muscle fibers it controls, right? So every major muscle of your body has, I don't know how many, hundreds if not thousands of motor units within that muscle. And if each motor neuron has its own threshold, what would happen if we started to stimulate a muscle at a sub-threshold voltage and then slowly increased the voltage? How would the muscle respond? Well, I'm going to go to another diagram. Okay. I don't have the diagram here that I wanted to show. Not sure if I can show this on the screen or not. I need to be able to draw on a blank sheet. How do I do that? Not sure how to do this. Okay, I'm gonna try something. This probably won't work. Can you see me drawing on the single muscle twitch slide? Yes?
Yeah.
Okay. All right, so let's pretend that this line that I've drawn here. And these little, oh, they're not going to stay. Oh, shoot. Why is that doing that? They're coming off the line, aren't they? I'm not sure I can explain this without drawing it. And I can't seem to draw it here.
Did you want to draw it on a piece of paper and then just show it?
Yeah, I was thinking about that. Yeah, I guess that's what I'm going to have to try to do. Okay, so here's the scenario. We're going to deliver a number of different volts to that isolated gastrocnemius muscle of the frog, and we're going to see how the frog responds to that. Okay? Everybody see the scenario? And we're slowly increasing the voltage. We're going to start at 4 volts, and we're going to go up by 2 to 22. How will the muscle respond to that? I'm going to draw that in red pen. Okay, so here's how the muscle responds. So let's talk about what's happening here. At 4 volts, there's no muscle response. At 6 volts, there's no muscle response, right? Why? Because those are both sub-threshold voltages. What starts to happen at 8 volts? You can see the muscle begins to respond, right? And at 10 volts, what happens?
It responds a little bit more.
Right, exactly. And it continues to go up, doesn't it? Until we get to 16 volts. And then no matter how much higher we go, the magnitude of the contraction, the height of the contraction stays the same, right? So can you explain that?
So it reached its maximum threshold.
No.
It hasn't, yeah.
Well, yes and no. Think about related to motor units. At 4 and 6 volts, we said that was sub-threshold.
It's still being stimulated.
It's being stimulated the entire time, right? But at 6 volts, we've reached, I'm sorry, at 8 volts, at 8, the first time we see it react, at 8 volts, we've reached the threshold of some of the motor units. but not all of them. At 10 volts, we've reached more of those motor units. We've reached their thresholds. Have we reached all of the motor unit thresholds?
No.
No. At 12 volts, what's happened?
It's still increasing.
Yeah, I know, but explain it.
Closer.
I'm sorry?
You're reaching more of them
Exactly. We're reaching the threshold of more motor units.
when they just continue to say the same. That means you've reached all the motor units. then?
Exactly, exactly. Once we get to 16 volts and higher, we can say reasonably safely or reasonably accurately that we've reached the threshold of all of the motor units within that gastrocnemius muscle, and all of the muscle fibers that those motor neurons are connected to are contracting. Now, this process that you're seeing here, this phenomenon that I'm going to circle in green,
Please tell me it's not tetany.
this is called recruitment.
Oh, okay.
Recruitment. Recruitment of motor units. And you can actually see this data when you stimulate a muscle, like a frog gastrocnemius muscle. Or years ago, when I first started teaching at JCC, we did the frog stuff here like this, and then it got to a point where it was tough to keep the frogs alive, and you had to have a bunch of them, and the students were upset about you killing the frog. And so we ended up going with a different system that involved stimulating students' muscles. We had the, we had this device. We had this device that you would grab like a pistol.
Yeah.
And we'd stimulate this region of the inner arm.
So what year was this?
Oh, in the probably the early 90s.
So that's before tasers.
Probably.
You guys helped develop tasers.
Well, the story gets better. Just wait. And so when we stimulate this part of the arm, the middle finger would flex. Okay. So can you picture this?
Yeah.
So we got the student hooked up to this machine, little trigger device. We stimulate the muscle, and the middle finger would flex. And when every time the finger would flex, it would pull on the trigger, and that would record on the computer screen as a contraction, just like you would see here. You see a twitch, depending upon the paper speed, and so what we would do is we would start out at a sub-threshold voltage, and we wouldn't get any response at all. The finger would not twitch because we weren't activating or reaching the threshold of any of the motor units going to that muscle that controlled the middle finger flexion. But as we increased the voltage, this is exactly what happened until we got to a certain voltage, which if we met it or exceeded it, it didn't change the force of the flexion of the finger. It always was consistent. So we got this very same data that you see on the sheet here. Pretty interesting. So this is just delivering a single stimulus at a given voltage, but we changed the voltage as time went on. Another thing that we did was we delivered a single stimulus so we could see single muscle twitches. So here's a single muscle twitch. Let me get out of this. Okay, so a single muscle twitch is exactly what it says. It's delivering a single stimulus to the muscle, and this is how it typically responds to a single stimulus. And we can define what's called the latent period, and then there's a time of contraction and a time of relaxation. So what's happening during these different times? Well, During the latent period, this is the time between when the stimulus is applied and the time the muscle responds. There's a lag time there. It's a couple milliseconds, like, I don't know, 10 or 15 milliseconds. Well, what's happening during this latent period? Anybody know?
It's a delay. So like the stimulus is like delayed before like it can twitch.
Yeah, so what's happening physiologically during that time? Between when the muscle is stimulated and when you can actually see it begin to contract. Can you relate that to what we talked about in review yesterday? What has to happen between nerve impulse, conduction, and actual sliding filament theory starting to work?
Is sliding filament theory when the actin moves closer together?
Yeah, that's contraction.
Okay.
This is pre-contraction. This is the time between when the stimulus is applied and when the muscle first begins to contract. So there's no contraction during the latent period. But there's some things going on here. Think of the neuromuscular junction, right? The release of the acetylcholine across the cleft, the combining with receptor sites. Remember all this?
Mm-hmm.
The influx of sodium causing depolarization of the sarcolemma and the development of a muscle impulse that travels down the sarcolemma, goes into the T-tubules. All that happens during this latent period.
Okay.
What's happening during the time of contraction?
That's when the actin are moving closer together then?
Yeah, exactly.
Stroke, right?
Yep. This is the sliding filament model that we talked about. Those various steps in that table on page 302, right? Until we get full contraction of all the sarcomeres. What's happening during this period? Well, we know it's relaxation, but before that happens, what has to occur back at the neuromuscular junction?
That's when the ATP is there and it detaches from the actin sites.
Even before that, go back
That,
in time.
that other one, that long acetylcholine.
Oh, is that when it decomposes?
Yes.
Yeah, you've got to break down the acetylcholine at the cleft. So John's right on the money. The enzyme acetylcholinesterase degrades the acetylcholine. That prevents any additional muscle impulse generation, right? And if there's no muscle impulse generation, then what's going to start to be removed from the troponin and be inactively being pumped back into the SR?
The calcium.
Calcium. And if you remove the calcium from the troponin, what's going to happen to the tropomyosin? It's going to slide over, blocking the binding sites. Can the cross bridges form between the myosin and the actin?
No.
The answer is no. What's going to happen to the sarcomere?
It is open.
Going to elongate.
Then yeah.
That's called relaxation. So it's not just the relaxation, of the muscle. You have to go back and get rid of the acetylcholine at the neuromuscular junction to begin that whole process. All that's happening in the time of relaxation, which is why the time of relaxation is generally a little bit longer here. Why is that doing that? Why is this has to be longer because of that process that has to happen. Plus you have to actively pump the calcium back into the SR. That's much, a little bit more of a lengthy proposition or process than is the time of contraction back here. So anyway, we measured all this, we recorded all this data on students. It was very interesting. And then the last thing we did is we wanted to examine what effect does increasing the frequency of stimulation have on the muscle responding to that stimulation. So if you deliver a fairly low frequency stimulation like we're seeing on the screen here, you can see that there's ample time for the muscle fiber to, how do I get rid of this? There's ample time for the muscle fiber to relax here before the next stimulus causes it to contract. Okay? Everybody understand what I'm getting at? So you're delivering a low frequency of stimulation. What happens as you increase the frequency of stimulation? You shorten the time between when the muscle is relaxing and before you hit it again with another stimulus. Well, what we see is that the force of the contraction overall increases. That's what we're depicting on this slide. This is called summation. The magnitude of the individual twitches summate, they add together. The magnitude, the force increases. Why? Because we are decreasing the frequency of stimuli. We're not allowing the muscle to fully relax before we zap it again with another stimulus. If you continue to do that, if you continue to increase the frequency of stimuli, which means to decrease the time between stimuli, as noted by these vertical lines, you will first get summation, and then you'll get a point at which the muscle is in prolonged, sustained contraction. Because you're zapping it so fast, there's no time at all for a relaxation to occur. So back to our student hooked up to the gun device. We'd start off at a slow frequency of stimuli, and the muscle would twitch, like this, slowly. But then as we increase the frequency, it would start doing this. And then it would get to a point of high frequency stimuli, and the muscle would be like fully tensed up. You'd be pulling against that trigger. And then by that time, the student's kind of squirming in his seat and feeling a little uncomfortable. So then I turn the frequency up even more. And then they jump out of their chair. No, I never got to that point, but we did get to tetany. And when that happened, you could see it happen right in front of your eyes. It was quite amazing. Now, all this occurred within, I'd say, 20 seconds. So it just left a small burn mark on the arm. That's all. Not a big deal. No, wait, I'm just kidding.
So why don't we do this anymore?
What?
I said, so why don't we do this anymore?
Oh, I got it in the back room. You want to bring it out sometime? If you're having some depression, come in and I'll help you out.
I'll be over right across the hall. I'm just kidding.
No, I mean, it was really an interesting
guys-
lab. Go ahead, Jolene.
Sorry, I just real quick, Freya, I don't know if maybe she like fell asleep or something, but I'm just a little worried about her because she didn't like.
Come back up. Yeah.
Yeah, like I was like, thought maybe she was dozing and I didn't want to say anything and embarrass her, but now somebody was
She's
like,
gone.
yeah.
Yeah, she said that she had a seizure the other day. I think
Yeah,
before
that's why I was kind of
so
like,
I just want to make sure she was
She
okay.
like disappeared.
Yeah, she
I
had
just tried calling her.
she had one of these a couple weeks ago in lab, and I didn't know what was happening. But sometimes after you have a seizure like that, you're so wiped out, you just fall asleep like she's been doing, and I think that's what's happened.
okay.
Yeah, because she was just saying that.
Scared me a little bit when somebody asked me, so I just want to make sure.
Yeah.
Ooh.
Yeah, are you there?
I just tried calling her phone.
I don't know. You have her number, huh?
Yeah.
Yeah, we have a group
Got a
chat.
group
Oh,
chat
okay,
thing.
Yeah.
No, I think she's waking up. I saw the camera move. Freya, are you awake? No, I don't know what's going on. So those are some properties related to skeletal muscle physiology. I will certainly look into the issue of the audio not working properly. I'm not sure what's going on.
Ow.
They're calling your name.
Ow.
They're calling your name on the computer.
No shit. I was just.
Freya, are you okay?
Kinda.
Do you need me to call an ambulance or something?
No. I'm trying to research this. The rest of this.
I'm sorry, I got a train going by. I can't hear you.
Oh, all right. All right.
Do you have somebody there with you?
Yeah. Oh, yeah. Yeah, I don't want anyone else to know I had a seizure.
Well, I think everybody kind of knows something's up.
I know.
So are you okay?
Yeah, my husband's here. It makes me want to cry. Yeah, I'm sorry about yesterday. I just was like, I guess
But we're still in the classroom here, so
yeah, I know.
if you're okay, then that's all we want to know.
Yeah, I'm all right.
All right, maybe you'd better bow out and take a nap.
No, it's all right. Yeah, I have all this information here. I did want to ask you a question later, I guess, if after class, if I could talk to you.
Okay, I've got several people I'm meeting with, Jolene at 2:30 and then Myve at 2:45. If you want to pop in at 3 o'clock, I could meet with you on the Zoom room if you want to do that.
Sure.
Okay,
That's great.
okay.
Great. Thanks.
So we got 10 minutes left or so. Other questions on the muscular system?
As far as the threshold stimulus, does that have like a is like an amount of time that wears out?
No. The concept of threshold stimulus is just the minimum stimulus needed to elicit a response.
Oh, okay.
That's when you have a sub-threshold, you're below that magic value, whatever it is, voltage. And if you stimulate the muscle, for example, as we talked about with
Right.
the stimulator at 4 or 6 volts, it doesn't elicit any response because it hasn't reached the threshold of the motor units.
Oh.
When you start to recruit motor units by reaching their thresholds, the effect is to increase the magnitude of the contractions, like you see on the diagram.
Right.
Once
Okay.
you've gotten, in this example, to 16 volts or higher, the reason the magnitude doesn't get any higher, and when I say magnitude, I mean the muscle contraction magnitude, is because there are no more muscle fibers to contract because you're contracting all of them because you reached the threshold of all the motor units.
Okay. Yeah, like the optimum. The optimum.
The optimal number, the maximum number of muscle fibers are contracting
Okay.
at 16 volts plus.
Okay.
And every muscle of your body, every skeletal muscle, is composed of numerous motor units of varying thresholds. Now, ordinarily, when we're working, when we're walking, taking notes, doing stuff we do all day long, that involves contraction of muscles, and we're doing that right now as we speak, whether it's chewing, you know, a lunch like John is, or whatever the case may be, we are sending a sufficient stimulus via our nervous system to our motor units, and those corresponding muscle fibers are contracting.
And is that the partial tetanic contractions that you're just talking about with like everyday activities?
Okay, so anytime we talk about tetany, you know, full tetany, like I showed you on this slide, or partial tetany, like you're describing, which I did talk about, and the book talks about, like when you walk, when you sit in a chair without a back on the chair, you are inducing partial tetany in your leg muscles if you're walking, in your back muscles if you're sitting on the bar stool, or whatever the case may be. And the key there is understanding that it's partial tetany. In other words, some of the muscle fibers are being stimulated so quickly that they're in full contraction. If we had full tetany too long, that's going to induce fatigue in the muscle. It's going to induce problems in the muscle where you're going to build up an oxygen debt. Lactic acid is going to build up. It's not something that you want to, you know, necessarily do. But partial tetany means some of the muscle fibers are in full contraction because they're being stimulated so fast. But then a certain point in time comes when others become stimulated and the previous ones aren't. So you don't build up fatigue so much so quickly in those scenarios. But that doesn't mean, if I told you, you got to sit on that bar stool for 12 hours and you can't move, do you think you'd have a sore back at the end of, you know, 2 hours? Well, of course you would, because, you know, normally you don't sit in a, you know, on a chair for an extended period of time. It's, you know, you're not doing things that induce tetany typically in muscles. But if you're walking or sitting or whatever for short periods of time, it's not negatively impacting the physiology. So partial tetany is not at all uncommon, but we don't see muscles in that state for very long a period of time. So that's why they don't fatigue. I don't know if that makes sense.
Yeah.
Okay.
Thank you.
Yeah. Any other questions? Okay, I'll stop there then, and I will, I'm recording this. I will post this to the course shell. I will try to go in and see why there's the problem. There is. I can't promise I'm going to be able to fix it, but I will certainly look into that. And if you encounter other similar issues, tell me. Gosh, just the other day, I took a video that I recorded last spring and I edited it down for the microbiology class, and I had it set up. It was working fine. And then just a couple of days ago, a student emailed me and said, things are all screwed up. So I had to go back and play with it more, and I got it fixed. So I don't know if it's, you know, a YouTube end of the, you know, YouTube issue or what's going on here.
If you probably just made a note to just watch this, if you just edited this a little bit, I think this would be fine too.
Yeah, I was thinking about that. This wasn't my best Zoom lecture either, but I guess we got through it with my wonderful artistic ability. Yeah, I think that's a very good point, and I will do that. If I can't fix it, I'll say, hey, reference the April 7th Zoom class. Great idea. Okay, so you guys have a great rest of the week. I'll meet with, I'm meeting with Jolene now. Actually, Jolene, I'm going to pop out of this room to start the recording, and then I'll come back into it in about 5 minutes or less. How's that? Okay. Okay, see you guys later.